So what if I told you that cancer cells break the rules of metabolism? They prefer burning sugar the wrong way, even when oxygen is available. This weird phenomenon is called the Warburg Effect, and it might just hold the key to treating cancer. So in healthy cells, glucose is broken down to pyruvate, 6 carbons to a 3-carbon sugar, right? And then we obtain ATP, or adenosine triphosphate. We get 2 ATP for every molecule of glucose that comes out of glycolysis. We then take pyruvate to the Krebs cycle, or the tricarboxylic acid cycle. Pyruvate enters the mitochondria where the Krebs cycle takes place. We have energy carriers such as NADH and FADH2. These are reduced coenzymes that then are shuttled to the electron transport chain, and that's where we get massive ATP production, usually on the order of about 32 to 38 ATP molecules via the F0, F1 ATP synthase. Normally, cells prefer this high energy, high efficiency route because who wouldn't want more energy, right? But cancer cells play by slightly different rules. When oxygen's plentiful, they favor a less efficient pathway. They dump pyruvate into lactate, or lactic acid. There's lactic acid, instead of burning it for maximum energy. Normally, this is an anaerobic process. In the case of cancer cells, they do this in the presence of oxygen and high oxygen conditions. So cancer cells increase aerobic glycolysis. Even in the presence of oxygen, they undergo glycolysis followed by lactic acid formation, which is normally an anaerobic process. This also means they get just 2 ATP for glucose, but they burn glucose at a much faster rate. So where does it go? Why do cancer cells choose the low energy route instead of the higher energy, more efficient route? Turns out they're not after ATP. They're actually after something much, much bigger, right? Cancer isn't just about energy. It's about growth. And glycolysis provides all the raw materials for that. First, it's about biosynthesis. Faster glycolysis generates building blocks for DNA, lipids, and proteins. Secondly, it's about avoiding cell death or apoptosis. Mitochondria trigger apoptosis. The less mitochondrial activity, the better their chances of survival. So they're trying to stay in the glycolytic pathway, which is occurring in the cytoplasm. Third, there's signaling advantages. There's acidic lactate environments for both tumor invasion as well as immune evasion. But how do cancer cells rewire their metabolism? The secret lies in genetic mutations. First of all is the MYC oncogene. So the MYC oncogene boosts glucose uptake by cells, and glycolytic enzymes are transcriptionally upregulated by directly activating those genes that encode for enzymes in the glycolytic pathway, such as hexokinase and glucokinase. Number 2 is HIF-1 alpha. That's hypoxia-inducible factor 1 alpha. So this is a transcription factor that increases glycolysis and decreases mitochondrial activity in response to low oxygen levels. Cancer cells exploit this protein by upregulating it even under oxygen-rich conditions. Third is pyruvate kinase M2. This is an enzyme that catalyzes the final step of glycolysis, converting phosphoenolpyruvate into pyruvate. This is also one of the key regulatory steps in glycolysis as well. In rapidly dividing cancer cells, it is associated with its ability to function as a protein kinase beyond its metabolic role in glycolysis. This influences gene expression and cell proliferation. Cancer cells exploit this to show the final step of glycolysis, and this shifts the normal respiratory chain to lactate production and drives the cell away from energy and towards biosynthesis. Fourth is P53. This is a tumor suppressor protein that regulates glycolysis in multiple ways by slowing it down, promoting oxidative phosphorylation, and regulating the genes involved in glycolysis. Normally, P53 transcriptionally activates Parkin. This represses glycolysis by activating it, and that reduces mitochondrial respiration. Transcriptionally, we also activate TIGAR, T-I-G-A-R. That reduces glycolysis by degrading fructose 2,6-bisphosphate, which is an allosteric modulator of phosphofructokinase. So P53 transcriptionally represses phosphofructokinase B3 and B4, reducing glycolysis by reducing intracellular levels of fructose 2,6-bisphosphate. P53 also inhibits the pentose phosphate pathway. This suppresses glucose consumption by binding to glucose 6-phosphate dehydrogenase. And we also suppress the transcription of monocarboxylate transporter 1, MCT1. This inhibits the transcription of lactate in and out of cancer cells. So P53, when it's mutated, has the opposite effect of this and gives cancer cells everything they need for uncontrolled cell growth. So if cancer depends on glycolysis, can we block it to stop tumors? Scientists are working on just that. Several potential treatments are targeting the Warburg effect, right? First of all is glycolysis inhibitors, such as 2-deoxyglucose. This actually binds to hexokinase, and it acts as a competitive inhibitor of glucose metabolism and glycolysis. Second is lactate transport inhibitors, MCT1 and MCT4 inhibitors. So this blocks the transport of lactase into and out of the cancer cell, so it stops lactate from building up, slowing tumor growth. Third is dietary approaches. There's some research that suggests that a ketogenic diet or fasting may starve tumors of glucose. However, this is ongoing research, and there's other research that suggests that this may not actually be a case. So don't just rush out for that ketogenic diet just yet. So the Warburg effect isn't just a quirk of cancer, it's a fundamental shift in metabolism that helps tumors grow and spread. By understanding how cancer hijacks glycolysis, we might be able to fight back with new metabolic therapies. So if you enjoyed this deep dive into the biochemistry and cancer metabolism, don't forget to like, subscribe, and hit the little bell for more science content, and I'll see you in the next episode.